Hello everyone, my name is Shawn Mullen. I teach plant science and biology at Newtown High School, and I'm currently working towards finishing the tail end of my graduate studies here at Southern. Today I'm going to talk to you about Parkinson's disease-associated mutant LRRK2 mediated inhibition of microRNA activity that is being antagonized by TRIM32. Let's start out with some background to help you through some of the acronyms and terminology involved in this study. LRRK2 stands for leucine-rich repeat kinase 2, which is a common Parkinson's disease-associated gene. MicroRNA-mediated translation repression refers to small chunks of RNA that are responsible for RNA silencing and translational gene expression. TRIM32 is an activator of microRNA-mediated translation repression. Argo2 is an Argonaut2 protein, which is required for RNA-mediated gene silencing by RISC. And RISC refers to RNA-induced silencing complexes. LET7A is a microRNA studied in this experiment. G2019S is a mutation on the LRRK2 gene, and it increases kinase activity, while the D2016A mutation is the kinase dead mutation. Some further background about this experiment. Parkinson's disease is the second most common neurodegenerative disorder. Recent studies suggest that Parkinson's disease might have a strong neurodevelopmental component, implying that deregulation during embryonic development could aid in the development of this disease. Of the 15 gene mutations associated with Parkinson's, LRRK2 is the best known and most studied. Oddly, Parkinson's-associated mutations in this particular gene only cause the disease in about 30% of the carriers. So this suggests that other genetic or environmental factors must contribute to the development of the disease. Recent studies on Drosophila brains determines that pathogenic LRRK2 inhibits microRNA-mediated translation repression. Also important, cell cycle regulators were affected by alterations in translational control, and these findings further support the idea that these genes play a significant role in the developmental processes, such as cell cycle control. These observations support the theory that pathogenic LRRK2, via the microRNA activity modifications, affect the cell cycle in neural stem cells and thereby alter the fate of these cells during development. In contrast, TRIM32, the neural cell fate determiner, is an activator of microRNA-mediated translation repression. TRIM32 belongs to TRIM-NHL protein family. This protein family has been implicated in such biological processes as developmental timing, cell cycle progression, transcriptional regulation, and apoptosis, which is normal death of cell. TRIM32 suppresses proliferation and induces neuronal differentiation in neuronal stem cells from embryonic and adult mouse brains. TRIM32 binds directly to the microRNA-associated proteins of the Argonaute family, leading to enhanced activity of specific microRNAs, including LET7A. Okay, so what did they do in these experiments and what did they learn? It has been previously demonstrated in Drosophila that LRRK2 interacts with Argonaute proteins. The researchers first wanted to confirm if this interaction is conserved in mammals as well. They transfected human embryonic kidney cells with plasmids for overexpression of certain gene mutations. They wanted to see if the interaction was dependent on either the G2 or the D2 mutations of LRRK2. And they compared the human embryonic kidney cells with the brain lysates from the mouse subjects. As you can see in figure 1b, downregulation of LRRK2 in brain lysates from mice 2, 5, and 7 months of age. This indicates that LRRK2 has an important role during development. In addition, via the IP assays with antibodies directed against either LRRK2 or ARGO2, the researchers were able to confirm an interaction between LRRK2, TRIM32, and ARGO2, in turn confirming the existence and importance of a complex in vitro and in vivio. Next, the researchers wanted to test whether this interaction was mutually dependent and whether pathogenic G2 gene mutations had any influence on this interaction. So they generated double transgenic mice that either expressed wild-type TRIM32 or a TRIM32 knockout. Additionally, these mice either expressed the wild-type LRRK2 or the G2 mutation. The researchers used mouse brain lysates to conduct immunoprecipitation assays with ARGO2 antibodies. Regardless of the presence or absence of TRIM32, the ARGO-LRRK2 interaction was robustly present, as you can see in figure 2a. They were able to determine that LRRK2 and TRIM32 bind to ARGO2 independently, but reported opposite effects on risk, implying that they might compete for binding on the ARGO2 protein. Figure 2b and 2c demonstrate an interaction between ARGO2 and TRIM32, as well as between ARGO2 and LRRK2. This proves that LRRK2 and TRIM32 bind to different domains on the ARGO2 protein. Now, this is where the researchers were able to see a feedback loop. After proving that there was no binding antagonism between TRIM32 and LRRK2, the researchers tested whether there could be a functional antagonism involving microRNA activity regulation. Figure 3 shows that they were able to see an increase in microRNA activity in the presence of TRIM32, while the wild-type gene had no effect on LET7A activity. The expression of the G2 gene mutation as well as the R1 mutation significantly reduced the activity of LET7A. Co-expression of TRIM32 with either of these gene mutations on LRRK2 not only blocked microRNA inhibition, it also induced activation of LET7A, even if it was to a weaker degree. Also, a knockdown of TRIM32 inhibits the activity of LET7A. This combined data suggests a functional antagonism between TRIM32 and pathogenic LRRK2, and it also demonstrates that TRIM32 functions somewhere downstream of LRRK2. This is where the data starts to look really cool. The researchers cultured human neuroepithelial cells that were tagged with green fluorescent proteins and other neuronal markers. And they used Hotch staining to analyze pictonic nuclei, which are indicative of cell death. And this figure shows pathogenic LRRK2 inhibits TRIM32-induced neuronal differentiation. Pathogenic LRRK2 antagonizes not only TRIM32-induced microRNA activity, but also neuronal differentiation. So what do these results tell us about the function of pathogenic LRRK2 in Parkinson's disease? The data supports the hypothesis that Parkinson's has a strong neurodevelopmental component. Pathogenic LRRK2 inhibits the activity of microRNAs in neuronal cells during brain development and therefore interfere with precise cell cycle signaling and timing of neuronal differentiation. Correctly timed cell death is important in brain development. Disrupting properly timed cell death and developmental cycles can lead to altered neuronal networking, which can lead to higher likelihood of neuronal degeneration at later stages of the disease. I hope you've enjoyed this presentation on Parkinson's disease-associated gene mutations. Thank you for listening.